Okay. Perfect.
You can use this one. Leave this one.
Which one also? The video, yes. Okay.
Okay. Okay, so over the past 40 years, there's been many emerging and re-emerging diseases across the world, not just in Africa, but everywhere. You know, we would like to know what the reason is for this, but most of these were actually vector-borne and zoonotic of nature. So a couple of factors that we know is associated with the emergence of diseases include the alteration in contact rates between humans, domestic animals, and wildlife. So we think about agricultural expansion into pristine rainforests such as Nipah virus and Ebola that keeps emerging in Africa. The wildlife trade, the wet market, bushmeat, and export of wildlife, so which is associated with the emergence probably of COF1, 2, and 1 and 2, and monkeypox. Then global travel, with the increase in global travel, diseases such as SARS-CoV-1 and 2, pandemic influenza A 2009, as well as HIV, have rapidly spread internationally. The contact rates between farmers and poultry or livestock and wildlife, such as avian flu and Rift Valley fever. Then climatic events, the creation of new habitats, building of dams, El Ninos, and floods, increase the rate of vector-borne diseases, and then introduction of new pathogens and vectors in new regions such as West Nile, Zika, and chikungunya. And then, of course, global warming that allow insect vectors such as mosquitoes to invade new areas, including Aedes aegypti and Aedes albopictus that's associated with the expansion of chikungunya and dengue. And then animals as well that move to new habitats due to increase in temperatures. So before the pandemic, the WHO had actually come up with a list of priority pathogens, which included Crimean Congo, Ebola, Lassa, Middle Eastern respiratory syndromes, Nipah, and Rift Valley fever, and Zika, as the example of the important viruses to focus research because of potential epidemic and pandemic expansion. But they also included disease X. And so disease X is the knowledge or the recognition that there will probably be a new virus coming along, which turned out to be COVID-19. And that basically is now also on their list. But this list is currently being updated to expand it to more families that could be disease X. So I'm the scientific, or the chair of the Scientific Advisory Group for the Origins of Novel Pathogens, which includes COVID-19. But one of our terms of reference was to come up with a global framework that will give everybody guidelines on how to investigate the origins of novel pathogens. So this will be published soon. But in this we included viruses that are of high risk. So these include the hemorrhagic fevers, such as the filoviruses and arenoviruses, the arboviruses, and that mainly are the families Flaviviridae, the alphaviruses, and bunnioviralis. Then the pox viruses, smallpox, monkeypox, and other animal poxes. Zoonotic paramyxoviruses, such as the Nipah viruses, influenza A, that's always the biggest risk of a pandemic, and this includes swine influenza viruses, avian, but also any other novel influenza virus that acquires the ability to infect people. Then the coronaviruses, we've seen already 2 emerging or 3 emerging over the last 20 years, and there will likely be more. And then bacterial pathogens of concern that includes emerging and re-emerging zoonotic bacteria and drug-resistant bacteria. This may include anthrax, plague, and cholera. Okay, so in our framework, so it will be published in the next month. So you're getting a quick view of what we're about to publish. We included certain elements that should be investigated when you look at new diseases. So during early investigations, countries should have the capacity to have multidisciplinary teams of national and international experts that can do outbreak investigation, collect epidemiological data, sample collection, and then the ability to either transport it to be tested or stored, and development of diagnostic assays. During investigation of human epidemiology studies, countries should already have surveillance systems in place that can be used to retrospectively go and look at the potential of this pathogen emerging, collect epidemiological data, and have good diagnostic testing for common viruses, but also then for new viruses. Studies at the human and animal interface include then the capacity to do epidemiological studies of animals, which many countries lack. In environmental and ecological studies, so environmental systems such as wastewater surveillance or vector surveillance, and the lab capacity to test that. And then it's very important to have the capacity to either access genomic or phylogenetic laboratories with advanced gene sequencing expertise, where we can look at the phylogenetic relationship of a new pathogen to previous pathogens or precursor viruses to see where it comes from. And then biosafety and biosecurity control needs to be in place so that we can also be sure that it didn't accidentally escape from a lab. So this all has to do with the international health regulations. So countries needs to be able to show that they have multidisciplinary teams, have the capacity to do One Health research and surveillance to detect zoonotic viruses, and then lab capacity, diagnostic and biosafety capacity. So this brings us back then to the One Health approach. So the One Health approach, this is an old paper already that was published in, I think in 2012, but it emphasizes the importance of animal, human, and environmental health that should work together to enhance health for everybody. And the idea behind this is that we know that these pathogens circulate in the environment, and if we have the capacity to forecast outbreaks, we can reduce the outbreaks in animals, but you need to be able to then also detect it in animals and detect it in humans, so that then we can have a rapid response to reduce the outbreak in humans. And so recently the quadripartite has signed an agreement that's called the One Health Joint Plan of Action of working together for the health of humans, animals, plants, and environment. So the One Health approach has become more and more important. And so you can go and look at some of the references there if you have time. So how do we prepare then for zoonotic disease X? So the main ones are insect-transmitted viruses. We need to be able to do vector studies, but also the animal studies associated with that. A zoonotic transmission of using bodily fluids, so pathogens that are transmitted by rodents or bats. We need to be able to do this surveillance, but also then detect that in people. And then respiratory surveillance. Although the original vector is often important, once it spills over to humans, it becomes a human disease, and we need to be able to detect that. And this can be done through syndromic surveillance as well. So if you have systems in place to look at febrile disease, viral hemorrhagic fevers, encephalitis, and respiratory surveillance, and also then detect outbreaks in animals such as abortion storms that may signal things like Rift Valley fever. So I want to move then to the One Health surveillance of vector-borne viruses as an example of doing One Health surveillance. So the vector-borne viruses, as I mentioned, the most important zoonotic ones falls in the genus is alpha viruses, the Flaviviridae, and then the Bunia viralis. And so the alpha viruses includes viruses such as chikungunya, Sinbis, and the Eastern, Western, and Venezuela encephalitis viruses. These viruses are either transmitted by Aedes or Fulex mosquitoes, in the case of Sinbis, and can be found in Africa, Europe, and Asia, but also in the Americas. Most of them are associated with polioarthritis, rash, and acute fever, but some can then also present with encephalitis. The flaviviruses, Zika virus, West Nile and Japanese encephalitis is associated with neurological signs, but also acute rash and fever, and can be found in these different continents. But they are also transmitted either by Aedes or Fulex mosquitoes, and it's important to understand that. I will explain later the difference. Then viruses such as yellow fever and dengue present with hemorrhagic signs, also transmitted by Aedes mosquitoes and found in areas where these mosquitoes are prevalent. The Bunia viralis is separated into a couple of families, but includes Rift Valley fever and Toscana virus that's here in Italy, transmitted by either floodwater Aedes mosquitoes and sandflies, and associated with retinitis, encephalitis, hemorrhagic fever signs, or meningoencephalitis in the case of Toscana. And then Crimean Congo is the only one that's of these viruses that's transmitted by a hyaloma ticks, but it can be found all over Central Europe, Africa, Russia, Ukraine, and the Middle East, and is associated with hemorrhagic fever signs. And then the Ortobunja viruses, that's often less well known to the medical fraternity. But in Africa, we have one that's called Shuni virus, and Schmallenberg virus caused last outbreaks here in Europe around 2012 and onwards. And so these viruses was unclear where it comes from, but they are transmitted either by mosquitoes or Culicoides vectors. They're common in Africa and likely emerged from Africa to Europe. And then they can cause neurological signs and abortion in livestock, but also actually neurological signs in humans. Oops, wrong way, sorry. Okay, so the African arboviruses had really been some of the most important emerging diseases over the past couple of decades. And so we know West Nile, Zika, and chikungunya all emerged initially in Central Africa, and from there it spread to Southern Africa, to Europe, and onwards to the Americas, and can now be found internationally. So arboviruses can be divided into two groups. So the first group is the arboviruses that use non-human primates as a reservoir host in a sylvatic cycle, and then endemic Aedes mosquitoes. So in a rainforest situation, Zika, yellow fever, and chikungunya will circulate between non-human primates and endemic Aedes mosquitoes, and then spill over to humans, where it will be picked up by Aedes aegypti and Aedes albopictus, and then cause large outbreaks in an urban setting. But humans is then the reservoir host. And so humans can spread these diseases internationally, and where the mosquitoes are prevalent can cause epidemic large outbreaks. So you can find these everywhere where you find the vector. So Aedes aegypti is the main vector, but it's found in the warmer areas, so the more tropical areas of the world. And then Aedes albopictus picked up these viruses and had now become expanded into Europe. So you can get chikungunya now in Europe as well. It spread from Africa to India to Europe and onwards to the Americas, and expand the range of these viruses. So the WHO had formed this group, and I'm part of this as well. So the Technical Advisory Group for Arboviruses and the Global Arbovirus Initiative. And they really look at monitoring the risk, anticipate it, reduce the epidemic risk, strengthen vector control, prevent and prepare for pandemics, enhance innovation and approaches, and build a collaboration partner. But their main focus is the Aedes viruses. So we are trying to convince them to look at the Culex viruses as well. But at the moment, they basically monitor where you can find these different viruses and look at countries where you can get circulation of both. So here you can see Italy and South Africa comes up for chikungunya. And so chikungunya is actually not endemic in South Africa. We don't have enough Aedes mosquitoes to spread it, and we get occasional outbreaks through a sylvatic cycle. And I think Italy may be similar, but because you have Aedes albopictus, you get actual outbreaks as well. Okay, so the second group of viruses is arboviruses that spread with birds, ruminants, and wildlife that they use as reservoirs, and then circulate between these animals and either it's floodwater Aedes, so it's not the same as Aedes aegypti and Aedes albopictus, and Culex mosquitoes. So, for example, Rift Valley fever and viruses such as Wessels-Brand virus in Middelburg circulate between wildlife and ruminants and Aedes mosquitoes, and then can occasionally spill over to humans, where it will cause outbreaks. When people come into contact with abortions, it causes abortion storms, and also other animal products such as raw milk. West Nile, Sinbis, and Lesotho virus, and then the Shuni virus likely as well, spread between Culex and birds. So they need the bird basically as the amplifying host. And then during floods and high rainfall, it will spill over into humans and other sensitive animals such as horses. And so, but humans and animal or horses are dead end hosts. It means they cannot spread it back to the mosquito. They don't have high enough virus in their blood. But this is where you can actually see the disease. So it's possible to do syndromic surveillance by looking at neurological cases in horses to trace West Nile outbreaks as well. Okay, so in Africa, Rift Valley fever is really at the moment restricted to Africa, but it did spill over into the Arabian Peninsula in 2000. And as you can see, it's quite close to Europe, so it's a concern to you, but it's the reason that it doesn't go further is likely because it has a reservoir host in wildlife and in ruminants, and you can control it to some extent through control of animal exports and so on. And so between Africa and Saudi Arabia, there's been exports, and that's likely how it ended there. And if you have the competent vector, it will then spread. West Nile is very much underreported in Africa, and so, but you can find it in countries where that reported outbreaks that you see in red, but all the other countries actually have zero evidence that it's there, but it's not reported. So, but West Nile had really spread internationally, and so there's two major lineages, lineage 1 and lineage 2, and lineage 1 was found is the one that spread to the Americas and through Europe and Asia, while lineage 2 was restricted to Southern Africa and Madagascar. But then in the mid-2000s, it actually emerged in Central Europe, from where it now actually spread to the rest of Europe. And we know now that it actually caused as significant neurological signs as lineage 1. And so what's the reason for this? Why does Rift Valley fever stay where it is and West Nile go all over the place? And so we know that there's bird fly routes between Africa and Europe and Asia that every year gives it the opportunity to spread. And so these routes come through the Middle East, through Spain, and then also through Italy and Tunisia, and spread all the way down to Southern Africa. And so I told someone there's a picture of a bird that was ringed here in South Africa, and 40 days later it was found in Finland. So it has the capacity to very rapidly spread diseases. And so recently there was a paper by Mentzetali, if I pronounce that correctly, that actually shows some of the lineages that is associated between Europe and Africa. And as you can see, it's actually similar to the bird fly routes then as well. Okay, so in my group, we have a One Health program for many years where we looked for diseases in sentinel animals, so bird fatalities, neurological signs in horses, livestock and wildlife, and then abortion in young animals, or VHF signs. And then when we used genus-specific PCRs and serology, as well as next-gen sequencing to look for cases. And in areas where we found it, we look in hospitals for febrile disease, arthralgia, rash, neurological signs, and also respiratory signs, and then do vector surveillance in areas where we see cases. So over a period of about 15 years, we could show that West Nile circulates every single year in horses. But with that, there are certain viruses that co-circulate. So very specifically Cindus that you also find all over the world. And then viruses such as this Shuni virus that we described as a problem that wasn't recognized before. Wesselsbrand is quite rare because it's similar to Rift Valley fever, only happens every couple of years. And then other viruses such as the alpha virus is Middelburg, that's specific to Africa. We don't know if it occurs elsewhere. Outbreaks is associated with rainfall. So years with a high rainfall, you'll get a large outbreak of cases afterwards, and then the community develops some immunity. We could use the horses to show a very distinct seasonality between January and July, with few cases happening the rest of the year, but it gives you the opportunity to vaccinate in this time to prevent outbreaks in the time of vector activity. So in horses, West Nile had a high fatality rate of 36%, Shuni even higher, but it's more rare than West Nile. And then Cindus, we also detected about almost 30% fatality rate. And then some of the equine, this is equine encephalosis virus, it's an Orbi virus that spreads with African horse sickness as well. But all of them show neurological signs. And these are all photos of horses that we had in South Africa that died of Shuni, Middelburg, and West Nile. So we also get cases from wildlife vets, and so it helped us to describe the host range. So rhinos is almost a horse. They get similar diseases to horses, and we saw a lot of paralysis due to Shuni and Middelburg in them. This Middelburg virus, this alpha virus, causes arthritis as well, but also severe neurological disease in endemic cattle and also in sable. And then the flaviviruses we detected West Nile in exotic birds. So this is a South American flamingo that had an outbreak, but it doesn't kill the local flamingos, which is quite interesting. And then encephalitis in giraffe and various other animals. And then novel flaviviruses such as Begasa virus that we detect in the exotic birds. So in South Africa, our cases were mainly lineage 2, but we did detect a few lineage 1 cases in a lion in the Kruger Park, as well as horses in the Western Cape, showing the sensitivity of the system. And then there you can see the Begasa virus. So when we do whole genome sequencing, all these strains were from Europe. So you can see there's a very close relationship between the lineage 1 and 2 strains. And then these are lineage 1 strains from Europe as well. Okay, then Middelburg and Sindus virus. Sorry. The strains are similar to what we find in humans, vectors, and animals for Middelburg. And then Sindus is down here. And it's similar to strains that you find in Sweden as well. And then Shuni virus. So Shuni, remember, is the same family as Toscana, and well, not really, no, it's Toscana, yeah. But anyway, we find it in various different species. It has a very wide host range and cause severe neurological signs. But it's similar to strains that was originally identified in Nigeria. And then it emerged in Israel a couple of years ago, and it causes abortions and neurological disease there as well. And so it's something to look out for. And so we think it's likely spread by birds. And so in the last 2 years, we also then identified the Shamonda virus in a goat fetus, abortion. And what's interesting is there's Smallenberg, there's Shamonda. It's almost the same virus. And so we also then see cases of reassortment of between Shuni virus and Shamonda that will give rise to new viruses. So just human surveillance, when we screened CSF, we could show in hospitals that 3.6% of cases were West Nile, Shuni was 5.4%, and Middelburg 2%, but we didn't see Sindus. But if you do febrile disease surveillance, so acute febrile disease with or without neurological signs in hospitals, we could show that up to 8.6% average, which then ranged some years as higher, were West Nile cases. And of those West Nile, 64% had neurological signs that included febrile convulsions, encephalitis, and seizures. And a large portion of those were children. And so children is likely missed as a neurological signs, fever and rash in a child, as no one thinks of arboviruses. But then when we looked for Sindus as well, which was really interesting, 12% of the cases in hospital during the arbovirus season was actually Sindus. And it also, a child with a rash and a fever is never thought of an arbovirus. And a large number actually had neurological signs. So it may be inflammatory response that causes it, and it may not be neuroinvasive. We also then found Shuni in hospitals. Mostly, we did find it in adults with meningoencephalitis, but then in children with convulsions and fever. And then also in a child that was 13 days old, meaning that it never left the hospital and is likely maternal transmission. Okay, and then this is my last slide. So we do vector surveillance in areas where we found cases. So you can see these were the horse and the wildlife farms. And we detected West Nile and Begasa on many culex, different species of culex, but mainly inventatas and pipiens, which you also find in Europe. The alpha viruses also Sindus circulate with West Nile. It's in culex inventatus as well, and various other species. And then Shuni virus we identified in various Aedes mosquitoes, so floodwater Aedes, but also in some culex mosquitoes. And then importantly, we also find it in Culicoides. And so Culicoides is an amplifying vector. Basically, it infects large numbers of animals, and then probably mosquitoes bite the animals and then onwards to humans. Okay, so to conclude, many factors influence the emergence and re-emergence of infectious diseases. Climate change, global warming, industrialization, and globalization may drive some of the factors that increase the risk of spillover to humans and subsequent epidemic expansion. Zoonotic pathogens that acquire the ability to spread between humans directly or through common vectors have the highest risk of epidemics in new regions and pandemics. Vector-borne viruses that spread over borders through humans, birds, or mosquitoes at a high risk of international emergence, including to Europe and Africa. or between Europe and Africa. And then a One Health approach in humans, animals, and environment. Environment is needed to detect, prevent, and control emerging and re-emerging diseases. And then this goes with the improved syndromic, environmental, and genomic surveillance systems, diagnostic assays, which is often the limitation of detecting emerging arboviruses, and climate data that will enhance our risk to predict neglected tropical diseases as well as vector-borne diseases, and could, which we know will actually increase in the face of climate change. Thank you. And so this is my group, many students, entomologists and virologists and veterinary pathologists. They're not my students, they're my colleagues that's involved in this, and also then the infectious disease specialists at the hospitals. I've had funding from various sources, CDC, the National Research Foundation. LEARN was actually an EU grant, and then also from the GDD, G7, BMF, and then the Bill and Melinda Gates Foundation. And then just lastly, we have the One Health Conference in South Africa, the World One Health Conference in September. And so you must please attend, the largest One Health Conference ever. They received over 1,500 abstracts, and we promise it's going to be very exciting. Thank you.
Okay, thank you, Professor Brandler. That was really, really interesting, and thank you again for making it to this small.
So question at the end.
Yes.
Okay.
So are you already?
No, I don't know.
No. We do it now. Yes. We do it after what each talk. So I was going there. So if there is any question from the audience for Professor Bender, if you want, I can start. First of all, I'm really happy about your talk, because I think that you really touch many critical points in the global control of infectious diseases. And we know, like, each country should like, I see, the point is that I see the amount of amazing work in South Africa. And like, I think that, of course, as you said it, like, for a global control of this type of disease, every country should like create special and dedicated policy to support different, actions in order to control the diseases. And I'm very happy because you mentioned surveillance and also genetic surveillance, which is something most of us in the Mediterranean area do without any support from like our country or our countries. By using money, we like kind of steal from other projects, and how we do it because we recognize the importance and we know how important it is to compare genetically different situations, and in order to make
the screen without
how can I say, possible scenarios and understand what is going on. So I'm really happy you stressed this, because I think it's very important, and I think everybody agree with us. Do you think like the World Health Organization could like, or is going to like push a bit, trying to, work with different countries to push them to create policy, because now we don't have them in many countries. What do you think?
I think so WHO can push for policy, but eventually the countries will have to fund it. So South Africa will fund it. So a lot of the work that we do comes from international grants. The policy is written in place, so they support the work that we do it, but there's not always funding for it. WHO doesn't have funding. It comes from either the Gates Foundation.
But we think it have an impact on the decision.
I think so. So WHO actually has another program at the moment that specifically pathogen genomic surveillance networks.
Yeah.
And so you can apply to become part of that. I've applied for my lab, but it's normally government labs that's part, but they do say academic labs as well.
Yes.
And so it's something to look at.
Yes. If there are no questions, I have another. But I don't want to like, do you have questions? Any question, Alessandra? Maybe no?
We put them.
Well,
We discussed it.
yeah, no, I haven't done them. No, I forgot. I'm sorry, I'm a disaster.
It's not the report.
No, the point is like, so you like, do you think like, well, we all say like, we are waiting for the next bug, which will, you know, appear. I was like, thinking about like coronavirus, and more or less we have every 10 years, like, outbreak of corona, like we have SARS, then we have MERS, then we have SARS-2, and if you look at this, it's like every 10 years, which is a reasonable time. You know, Alessandra and I did some thinking about that, to create like, successful viral variants to go around and do what they did. But I think that even if like, I wanted an opinion, from you about One Health and the concept of One Health, which everybody talk about, but I think still is not properly declined, especially again in the policy, in the, you know, by the governments, like general countries, governments and local government. Because if we see, like, even if we have this pandemic, which caused, I don't know, like horrible damage everywhere, still, for instance, I'm talking about our country, I don't think we have like an effective, or we are thinking about an effective surveillance program, or we are declining one end as we can. We saw many people like communicating, like, and confusing other people, but actually that was a great occasion to apply the One Health concept. I don't know if you agree, Alessandra, I take the responsibility
Yes, I will
for
tell you
this.
after that.
But I don't think like, is effectively declined. It's a nice idea, but still we have to work on that. What do you think?
I think it's true, and I think that, so the vets like the idea of One Health, but often what we see in our agency is One Health, but it's for vets. And then there's One Health for humans. And it's really the whole point of this is that you need the three groups to work together.
Yes.
To enhance it.
Definitely.
And we unfortunately we see the same thing, but at the moment I think it's a good time to push for it. Even though I do see a lot of the epidemic and pandemic previous plans have One Health as a name, but then they go back and have to focus on TB and HIV and so much. That's the most important diseases for them. So I think it's the pandemic, like COVID and also influenza, which may be the next one again. We know it will be comes every 20 years. But it's a reminder why it's important to search or to do surveillance in animals and humans and then the environment as well. And there's a lot of new systems in place. I'm quite impressed with wastewater surveillance. I think it's something that we've missed over the years, and it's a way to do population surveillance in a larger scale than detect what is circulating. And it's amazing what you can detect in wastewater, anything from respiratory viruses to other viruses as well. So the problem with surveillance is expensive. And so it's important to, I think sentinel surveillance is an important alternative to that.
That's a problem.
But wastewater gives you opportunity to do population surveillance.
Yes, so thank you, really thank you. And if there are no more questions, we will go on with next speaker, Alessandra Scagliarini. She is also a biologist. She's from the University of Bologna. She's full professor at the University of Bologna, where she coordinates the One Health International Study Center of the University of Bologna. Alessandra made also a lot of work on One Health approach, and like she's the living proof of One Health, because she's a vet working with hospital, human hospital and medical doctors. So she's like, you know, applying the concept of One Health. And also in 2023, she received the One Health Award as a best Italian scientist in the field. And well, she did a lot of work at the European Commission level, like for this type of diseases. So Alessandra, I'm not going again to say everything about you, but we know, you know, I'll leave you the audience, and she's going to give us like talk about like the light, the bad and the light side of One Health approach management, especially I think in Italy, you will talk about that. So, well, and everywhere.
More or
like,
less.
but please, you have 20 minutes and then maybe 5-6 minutes for questions at the end of your talk. Okay?
Thank you,
Please.
thank you. Thank you, Alberto, for the introduction, and thank you for organizing this very nice meeting in a very nice place. I will try to do my best with the presentation in this mode. I hope it will work. So I did my own work because this assignment was just given from Alberto to me, and he said, do you agree to do that? Yes, I will try to do my best. So I will zoom out and I try not to overlap too much on what Marietjie already said. We work together, so it's quite straightforward that we do more or less the same things. But yes, so we are diving in the Anthropocene where the human hands had modified the environment so much so that somebody was estimating the quantity of the things that were built by humans compared to the ones that were built by nature. And now we are in an era where what was built by humans is much more than what was built by nature. So we have to cope with that because we have modified the environment. We are just having such huge modification that are able to modify the epidemiology and all that we know already about some diseases and the one that will come next, pathogen Higgs, for example. So I think that this slide, I love it because somebody was trying to estimate the overall amount of biomass on the planet, and over 550 gigatons of carbon that we have of living beings on the planet, 450 are plants. And so they are the huge amount of living beings on the planet. But I think that it's very interesting to see that microorganisms are also a lot. So we have a lot of bacteria. Not all of them are pathogen. They are so important everywhere in our body, in the body of the animals. So the microbiome, now we know a lot about the importance, but also we try to figure out, and we don't know much about the microbiome that is in the environment, like water and also ground. So in the environment, we have a lot of bacteria, which are so important for the growing of the plants, but they are also important when it comes to antimicrobial resistance and antibiotic resistance, because they are also able to transfer some of these genetic determinants. Then we have viruses. They are tiny, but they are also relevant. And then we have animals. So animals overall are 2 gigatons of carbon. Half of them are arthropods. So it makes a lot of sense when we talk about vector-borne diseases, especially in time of climate change. If we compare all the animals, we will see that humans are just, we are animals at the end. I'm a spillover, so I moved from the vet school 5 years ago to the human side. And when I teach at the medical school, students are so amazed about the fact that we are animals, we are mammals. And they're, oh, yes, it's true. Yes, we are mammals. So when you wear the eyewear of a microorganism, you don't see any difference. And then, but it's interesting to see that the main part of the animals on the planet are now livestock. So domestic animals, and especially the ones that we farm, are so huge amount. And they are much more than wild mammals. They are much more than wild birds. So when we blame wildlife for spilling over microorganisms to humans, then we have to pay attention. What is the real interface? So I will try to talk about this. So the other thing, it doesn't work because it's not, sorry, I want to show you like that. No. So we have livestock, wild mammals, humans, and then we neglect the microorganism, which are the main part. Just a very tiny percentage that we know is pathogen. But then we have these ones. So we have mosquitoes, we have ticks, we have other very interesting flies that are so important as vectors of diseases. So we have to cope with that. And when we talk about emerging infectious diseases, especially the ones that are transmitted through these animals, we need to really pay attention of environmental factors. I will skip again to the other way because I know that you will get angry if I'm not. No, because they said it doesn't work. It doesn't work in full screen. I don't know why. So yeah. So Marietjie already mentioned this. So about all the epidemics that we faced in the last 30 years. Of course, we are getting used to new discoveries because now we have a lot of tools to discover new pathogens. So probably we know how to discover them. But of course, we are changing the environment so much and our ecosystem are overlapping in a very strange way. We have globalization, we have traveling of people everywhere, and then intensive agriculture and so on. So it is not surprising that we are having this spillover from animals to humans. But I would like to say that we also have spillover from humans to animals because we are animals. So it's just interspecies transmission. And so we have a lot of these amazing viruses jumping from different animal species. And we don't have to forget that also AMR, it's a huge problem that is related. It's a zoonosis itself. And it's not just related to antibiotics, even if we know a lot about antibiotics, but because we are dealing with vector-borne diseases, we know that we also have to cope with antiparasitic that are not able to reduce these parasite population and antifungi and so on. So why is that? These are the main challenges that we are facing at the moment. So especially deforestation, I will talk about deforestation that is linked to farming. I said that we have a lot of livestock, we need to feed them. So we need pastures, we need to widen the environment where they can graze. And this is really causing the proximity of these pastures and an area where they feed and where we do our farming activities with forests that becoming less and less extended. And of course, if the population is expected to grow to 11 billion in 2100, so 21, it will be very, very interesting to see what will happen, because if we do work as usual, we have to reduce forests more and more, and we have to use more and more chemicals to produce enough food for everybody. But another very important factor is urbanization, because people are expected to live more and more in urban settings, especially in low and medium income countries where the growth of the population will be expected more. And these urban settings are expected to really, really spread into the wild or near or in proximity to the wild. And this already caused some modification in the spread of some diseases. Think about Ebola in 2014, where we had this urban spreading that was never reported before, because then it was a rural setting that was really characterizing this disease. So urban setting is also modifying the suburban areas where we have this fragmented ecosystem, where some animals are just disappearing, just like big mammals, like and predators. So we have a big concentration, for example, of rodents. And this is also modifying, for example, the ecology of some diseases like Lyme that we were expected to have into the forest, into the bush, but now we are having a lot of Lyme cases in suburban areas because of the concentration of rodents that are very happy to transmit Lyme disease to ticks, which are very concentrated there. And also in urban setting, if we think about Rome, if we think about Bologna, we have a lot of wildlife in the urban setting, like wild boars or deer that are just walking in the street because they just come from the bush that is not so far from there. And of course, what should appear after that is climate change. So the orange is climate change. So what we are just seeing is that it's more and more hot, rainy, very strange weather here in Italy. It's like being in a tropical country. So we are also coping with these different weather events that are just hitting us. I want to show you this because I think that this concept is very important. When we talk about infectious diseases, this is not just a biological fact. It's not just having a pathogen jumping inside a body, replicating and giving a disease. It's also a matter of how this pathogen jumps into human population or domestic population. And I think this concept is quite interesting. So the ecotone, what is the ecotone? Ecotone is like an intermediate area where you have agriculture, farming, and so it is an area that comes from urban and peri-urban to the wild. So this is a very interesting overlap where you can have the chance to meet wildlife, where you can have the chance to meet the ticks or the flies that normally stay in the wild. You have animals that are able to go through all the three areas like rodents. They are very happy to move. Others that are just going from the wild to the ecotone and maybe meet some livestock that are grazing there, or meet humans that are just going there for doing farming and so on. So characterizing the ecotones that can be very different in different areas. This is, I think, very crucial when it comes to managing zoonotic diseases, because we don't have only to take into consideration host and pathogen, but also where we have the chance to get these pathogens. So, of course, we know and we blame a lot wildlife, as I was saying before, but many times the real interface are domestic animals. So we have domestic animals, and this happens with Nipah virus. This happened with other diseases like, I don't know, Chiasanur, other emerging infectious diseases that have a reservoir in the wild, but they are able to jump into the human population thanks to an intermediate interface that is just represented by domestic animals. And of course, if we put vectors there, so if we deal with vector-borne diseases, we have another very big complex ecology that is the one of these arthropods that I said they are very, very relevant on this planet. They are just half of the animals that live on the planet. So indeed, it makes sense to manage all these with a One Health perspective. And the World Bank published a very interesting report in October 2022, showing that investing in prevention would be more worth that going to preparedness and response. And they estimated, well, how much we can save if we invest a lot in prevention. And the World Bank underlined the importance of really embracing a One Health approach, because this will be an investment for humanity's future, which is, you know, a very important part, because they have identified all the environmental factors that can be important and relevant when it comes to zoonotic diseases, and they identified a lot of knowledge gaps that we need to really focus on in our research. And they are funding research, so they are a funding body. So we have to take care of what they say, because maybe we can find also funding opportunities. Marietjie already mentioned the One Health Joint Plan of Action of the quadripartite, and this is a very important report, March 2022. For the first time, the FAO and WHO and WOA joined with UNEP. So now environment is there, and environment is so important now. And the tracks that they have identified deal with zoonotic diseases, and especially neglected diseases and zoonotic diseases, but also vector-borne diseases. So we know that we are now talking about something that is considered very relevant also to the quadripartite. And so management of zoonosis needs to take care of these three words: prevention, preparedness, and response, with a One Health perspective. And as we know, the concept of One Health is very dynamic. So from the first graphic that is there, the 2003, the Karish interview from the Washington Post, it evolved into the Lancet ones, and then in the more complex that was launched by the Netherlands One Health Center. That is quite interesting because you don't have just biology here. You have a lot of social science here, meaning that you cannot do One Health just in the medical sector or in biology, in biomedical sector, but you really need to understand which are the social factors that affect what we observe in our daily practice, because culture, food habits, religion, and also social factors like conflicts that we have all around us, but also migration and many other, but also tools like genomics is changing the face of what we know and what we will know very soon. Of course, we were not very happy about that because then the One Health high-level expert panel launched this new concept that takes care of the social part in particular, and the approach that needs to be transdisciplinary. And this panel is very active. These are the main, I think, pillars and the concept that they wanted to underline. So the equity on the sectors and disciplines. So when we talk about health, health is just an indicator, a prerequisite and an indicator of sustainable development. So it is not just a medical thing, especially if we focus on prevention. We have to cope with something that is not in the hospital. So when you are in the hospital, it's too late. So we have to do something well before. And then we have the socio-ecological equilibrium, but also the social-political. So we cannot do our science if we don't have policymakers with us, because they are also funders. And so we have to consider also this part, but also the disciplinary. So we need to work with communities, because sometimes doing epidemiology with the help of local communities is much straightforward, because they can just inform you of what is happening in the field. And sometimes they are also very engaged in participating. And this is also very important, because when you find solution, you need to have this solution accepted. We face this with vaccine for COVID-19. We had a beautiful vaccine, but then people didn't want to vaccinate. Why? What we have done wrong? Maybe we don't have worked with people. We just scare them. And so, of course, in terms of, well, we have social media and other things where people getting information, and maybe as a scientist are not very good to work with communities. And this is very important. So the One Health high-level expert panel worked a lot also on management of zoonotic diseases. And in the red there, you see the science part. In the blue, you have the management, because you cannot do risk management if you don't have risk assessment before. And risk assessment is based on identifying the data that are needed to really manage a complex system that we have already mentioned. To tackle a complex system, you need to have very different experts that understand the different factors that are around the phenomenon that you are observing. So it is quite interesting how to deal with step number 1 and 2, and then go to 3, 4, 5, and 6. So for step number 1 and 2, I said that we need to identify which are the main factors that are just around a complex dynamic that you are observing when, for example, you have an emerging infectious disease that may be zoonotic or not. And so you have to answer to all this question, but to answer to all this question, maybe a vet or a medical doctor is not enough. You need maybe an entomologist, you need maybe somebody that is well aware of what is happening in the field. Maybe you need a mathematician or a statistician or an engineer that explain what is happening. For example, wastewater surveillance, you need environmental engineer to understand if what you are sampling makes sense or not, and how to deal with the complexity of this kind of sampling. And then this is important because when you identify, well, maybe not all, but you are trying to discover all these variables, then you can design measures that make sense when you want to manage these kind of diseases. So we are dealing with social ecological system where we don't have just host and a pathogen and maybe a vector, but you have a lot of social dynamics inside, and you need to really design the system to understand where you are. So we try to do this exercise in the area where we work with the One Health Center. And this is, you cannot see, now I will move because you have to see that. So, all right, so this is Romagna. And Romagna is just 1/3 of the whole Emilia-Romagna region. And it's a very interesting area because the local population is not so big, but during the summer, you have 10 times the population. People are coming for tourism there. They come with their own animals and then they go. So whatever happens during the summer can be spread everywhere after the holidays. So this is one of the things that you need to think about. And then if you want to see the ecotone, so you have the urban areas here, then you have the two greens, they are national parks. So you have two national parks there. One is a wet area, a big wet area, so the Delta del Po. And then you have the Saline di Cerva, another wet area. And then you have the Parco Nazionale delle Foreste Casentinesi, where there are a lot of wildlife. And I don't know, these are the ecotones. So I want to show you what happens here. Thank you. So this is the ecotone. Okay, what happens here? So these are the two wet areas, and then you have all these animals. First of all, the farming things that happens in Romagna are very different from Emilia. So you have poultry, mainly poultry, a lot of poultry there. And then you have a lot of wildlife, maybe mammals and wild birds. And so you have to imagine ticks, and you have to imagine all the vectors that can be here. But then here you have a lot of birds, very interesting birds that are attracted by this wet area with all the insects. And yeah, last year we had a big flooding. So we really experienced what may happen during such an extreme event. And then we also have a biosymbiotic district that is a European district that is investing in enhancing the microbiome of the ground to improve farming practice and agriculture, and also livestock farming. So they are experimenting with a lot of research institute there to see if they can reduce the use of chemicals in that areas. Of course, if they reduce the chemicals, something will happen, because we are a very dynamic system. And then where we are, this is the system. So we have university, but we have all these other stakeholders. So when it comes to understanding what happens, you have to understand where you are and which are the important stakeholders that you have to deal with to understand and to manage for the best. And this is one of the systems that we depicted during a European project dealing with One Health. And yeah, I think it's more or less complete, but just to identify how many people you have to involve when you want to discuss about new management solution, and to understand which are the constraints related to sampling and so on. In the meantime, we had a lot of new things happening at the Commission. So now we have a One Health Directorate at the Commission. And so this deals with point number 4, developing a system governance, because you cannot do research and One Health without the support of the European Commission if you live in Europe. And now in the Horizon Europe, you see that many calls are dealing with One Health approach in cluster 1, in cluster 6, but also in the cluster that deals with climate change. And then we have this interagency new policy. So all the European agencies now are working together to establish a transversal way of working, because they realized that they were all focused on their own mission. So they were just neglecting or overlooking what was happening in the other agencies. And now they are working together to facilitate strategic coordination, promoting research coordination. They did an audit on Horizon 2020, and they noticed that, first of all, we have to involve not just Europeans, but also other countries in our project. Is not just one call, but all the calls can now involve also Africa, Asia, and so on. And also you need to involve social scientists also in your health calls or food and so on. So this is very important thing that happened. And now we have the Italy. So the National Plan for Prevention mentioned One Health, mentioned the environment is very focused on AMR. But of course, it was 2020, so pandemics, emergencies. We hope that will last after 2025, we will see. But that was adopted by, of course, my region, Emilia-Romagna. And we know that Emilia-Romagna is well advanced in a One Health surveillance system, for example, for vector-borne diseases, in leishmaniasis, in West Nile, chikungunya, Toscana, and many others. And so we are well advanced, and the thing is very strange because we have the same Istituto Zooprofilattico, is Institute for Animal Health, but for example, in Lombardia, that deals with the same Istituto Zooprofilattico, they don't do that. So it's very strange. It depends really on what the health sector in your region is dealing with. Going back to our system, this is again Romagna, and in Romagna we have different medical school in different provinces. We have a lot of health program spread everywhere. If you go to Emilia-Romagna, everything is in Bologna. If you go to Romagna, you have in each province something that deals with health. So it is quite straightforward to realize a nexus. And this is important when you want to deal with One Health surveillance, because everybody is engaged. It's not just somebody that is in one university dealing with a big grant, but everybody can share. And this is another very important part when you want to promote One Health. And this is just an example of what we are doing. For example, now we launched this big surveillance on fever of unknown origin. And we noticed that 50% of the patients are going back home from the hospital without any diagnosis. So 50% is like 2,000 in 5 years. So they go back and they had a fever for a long time, sometimes with neurological signs, sometimes not, but they didn't have any solution. And of course, the number of cases are quite linked to climate. So it is important to notice that, because probably we are overlooking a lot of pathogens that circulate during the summer season, but we don't know. So whole genome sequencing and all of that will help to understand what is happening. But also we have to educate the policymakers, because policymakers are not adapted to working in One Health, but policymakers are the ones that fund, are the ones that implement or help you to implement this kind of management, holistic management. What we do in our European Alliance, which is Una Europa, and is the only one in Europe having a One Health focus area. We are trying to really operationalize One Health through education, through research, and through community engagement. We launch a lot of research initiative, and Marietjie knows this because this is a diagram from one of our grants that we hope will be funded, but it's very recalling some of the concept that she was saying. So syndromic surveillance, sentinel screening, developing diagnostic that are just focused on rural areas where you have the ecotone, where the pathogens are jumpy. So you need very simple diagnostic solution for these areas that sometimes are so remote that they don't have whole genome sequencing facilities and so on. Working at the interface with wildlife, this is a big project on ticks, for example, and community engagement in Africa. Carla, I think, knows what I'm saying. And characterization of vectors. This is very important. We know a lot in Europe. We don't know so much in Africa. And this is why we are doing that. And educate policymakers. Educate policymakers. We are launching a lifelong learning on that because they need to know how to work in One Health. So this is all connected. And this is the group that contributed to this so far. Thank you for your attention.
Because we are running out of time, and there are a lot of stuff here. It's very important, especially for the networking, like mentioning all the organization, the joint organization, etc. So I would like to pass that stuff later on, because we are
Well,
running out.
now, if you want, we can have a coffee break,
Yes.
and then maybe you can stay 5 more minutes and we can.
Yeah, let's do one thing. We have a coffee break now, and then we come back here for some questions to Alessandra.
It's unorthodox.
And sorry, just the speakers. I will only allow 20 minutes. I will be very strict myself because I can see University of Sassari is very gentle.
Your sense.
I will see the advance. Now, considering we have next presentation is 1.4 GB and it is in PDF.
No, it's not 1.4. It's a Camilla.
Sì, sì, Camilla.
We are still in area 1.
Yeah, okay.
It's 1.4.
This,
I know it's a PDF. It's
this
fine.
one is PDF, so
Yeah, it's fine. We can do next one.